right, thank you.
Hey, thanks for having us today, Cassie. We're happy to present on this project. Um, so it started with Steve Gustafson, and he'll be speaking shortly. Just through some research of his own, he's the current chair of Partners of Scott County Watersheds. Let's see. So first, what is bioremediation? It's essentially the use of microorganisms to clean up contaminants. The image on the right isn't necessarily indicative of our project specifically, but I just like the simplicity of it. Take really dirty stuff and it comes out cleaner. So today we'll talk about what the filters are exactly, and how the install went. So just a little bit on the costs, and then case studies and data so far. And we've got a new project happening this summer that Kimberly will talk about in a little bit. So Steve, I didn't know if you wanted to provide a little background on Franklin County and how you kind of came up or found this information.
Sure. So I was back, I don't know, probably 6 or 7 years ago. I was really interested in looking into in situ, kind of in stream ways to deal with contaminants, E. coli or basically nitrate and phosphates. How I could use the stream to kind of treat itself. So I came across this presentation on the internet by Franklin Soil and Water Conservation District, where they had taken this material that basically, in one of the next slides, you'll see that they use floating water treatment islands made out of, and they cut it into strips and put it into streams. And they were basically looking at more creating kind of pools behind it to create habitat for small and invertebrates. And they noticed some decreases in E. coli. They were not a huge district. They didn't have a lot of budget to kind of monitor. They just kind of noticed it, put it in a presentation, kind of caught my eye. So that sparked kind of this interest into, wow, what's going on here? I kind of hijacked the idea, I guess I would say. They came up with a lot of experimentation of how to put these things in. You'll see this is one of their early installations. This next slide here, these are from theirs. You can see some of their failures on that left-hand upper corner there, like experimenting on how these things to put them in and to getting blown out during storms and such. So we kind of learned from them. I was able to talk to the guys that did that before he retired. They probably did it in 3 or 4 spots over a few years just to see what worked, what didn't. So I was able to learn quite a bit on installation and logistics of what worked and what didn't before Amy and I even got a chance to put these in ourselves. I
Yeah,
don't know what
they
your
did a
next
lot of
slide
the
is.
legwork there. So
Essentially, yeah. I don't know what your next slide is. And so, yeah, you can see on the right hand is the traditional use of this material. It essentially is a plastic mesh. I kind of equate it to like steel wool, but it's plastic, and I think it's recycled plastic, like from 2-liter bottles and such. And there's a bunch of different uh, companies that sell these. Biohaven, I think, is one of them, and we have these around different places in the Quad Cities. And what they're usually treated for is, like, you put plants in them, you put them out on a waterway, the roots go through, and they basically bring in nutrients, right? But you can buy the material in strips. I can't remember how, like, what, 100-foot long strips, I think, Amy, something like
yeah,
that.
it was it comes in like 7.5 foot tall by 90 lineal foot. So it's a giant roll. I got a image later in the presentation, but
Yeah.
yeah.
To do with what you want, right? And so it's this material which you see on the left hand side, which is basically what we want water to float through. So we're changing what was basically a matrix for plants to propagate through and they're to grow into, to basically change for a place for bacteria to habitat through. And we'll talk about that more with Kimberly, basically, and water to flow through. That's what Franklin Soil and Water Conservation District kind of created. So that's basically what we're doing. We're changing the purpose of material. And then Amy, I guess if you want to talk about more logistics of how we actually built these? Because
Yep.
that was a lot with your folks as well.
Yeah. Yeah. So the Franklin County was able to put together these images for what they learned in their install. On the bottom left, on one of the pictures we saw, the material kind of lifted up from the flow. So they found if they were to put kind of a blanket over top of their layers, that would kind of hold everything in place and prevent it from lifting. In addition, they found that if you created a depression in the middle of the layers, it would keep flow from hitting the banks. Also, it was a requirement, and the second set of filters we put in from the core. They had different plan reviewers from the first project to the second, and so they were just looking for some different types of things. But they did all of the legwork, so we had a pretty easy install. The anchoring is primarily through these duckbill anchors, where you slide them in, and when you pull it taut, they kind of spread out like a wall anchor. And that helped keep the stakes in place. And these are just concrete stakes that you can get from, anywhere that anybody that deals with concrete or setting up forms, that these will go into the ground. And then used wires to connect everything. And we've got another picture on that too. And at the first install, we actually had to get 4-foot anchors. We started with 2-foot, but the sub base and surrounding soils were so saturated that we couldn't really get any tooth on it, and they pulled out. So depending on what your environment is, the 2-foot stakes could be appropriate, but if it's really squishy, you may have to go to 4-foot long. And here's an image of one of the rolls, but we still have one in our with our supplies. So they're 7.5 feet tall by 90 lineal feet. We got it from a place in St. Paul called Brotex. And I think they also sell like the pre-made floating islands. So we had to sign an agreement that we wouldn't be using this material to make those, just so they can, protect that revenue source. So if you are thinking about that, you might just have to go the route of purchasing them pre-made. But the first project cost about $12,000, and that was with labor and equipment, and with the initial sampling and analysis. We did use an excavator with our projects to key in the sides because we were putting them on streams that had flows a little bit higher than the Franklin County. So they could be installed for much, much less, but having our equipment operators and equipment there definitely had those rise up. And then the tree removals, we were putting in the first project, and then the derecho happened. So we had a lot of removals that we hadn't planned for just to even access our project sites. So that probably won't be indicative of all projects either. So this is Davenport. For the most part, a little bit is cut off. And then our first project was at the Robin Creek Dog Park. And this is what the kind of the watershed looks like. It's mostly single family homes. There's some on the top left, larger 1 or 2 acre lots, but pretty well developed. We chose a spot since we didn't have much to go off of, and this was just kind of an experiment. We decided we'd go with 3 filters. We wanted to put them in on the straight sections so we didn't disrupt any of the sinuosity or have issues with additional bank erosion. And so it's a pretty small watershed. There's about 400 acres that drain to the project site. There are 2 really big storm pipes that come in at the top. There's a 78 and a 66-inch. So it's a super flashy system. When it's raining, those pipes are running pretty close to full. And then when it's not, there's a good amount of base flow, but it's probably around 6 to 8 inches, I think, is the normal flow. And this was the site that we needed to use the 4-foot stakes on instead of 2. So early install, like I said, we brought in equipment to key in the sides. I think the Franklin County initial filters were just installed with some rocks kind of weighting them down, but we wanted some extra assurance that they wouldn't move. Here you can see Mike and Steve in the water, getting muddy, putting the filters in. And I think that the initial information from Franklin County had the top piece that goes over the whole system kind of keyed in, but that is a challenge. And I don't think we actually keyed in across the bank on our second project because the duckbill anchors and all the wires were able to hold it in place enough. And here's the guys working. They have a washer on top and to help keep that in there. And then on this one, compared to the second project, you'll see the stakes were only up maybe less than an inch, maybe an inch. And I think the holes that the wire went through on the second project were just in a different location, and that has led to some different issues. And I think it was a pretty chilly day when we were working on those. It was hard to get the anchors, and everything was just really small. I remember getting all the tiny nuts attached to things was a challenge. So got to have some nimble fingers. We did get some signage put up at the first project site because we got some grant funding with Iowa American Water and from River Action. So just to provide some education, we do get calls now and again through the city asking what they are. This first project, you can't see them very much anymore because they've just accumulated sediment in plants and things. But when they're first installed, we did get quite a few calls. This is I think one of the first ones on the first install. On the left, you can see it a little bit better. On the right, it's got quite a bit of gunk covering it up. But we would sample upstream and downstream of the whole system. At first, we did up and downstream from each individual filter. But that's when we had some additional assistance and funding through St. Ambrose University doing some monitoring. When that dried up, we just moved to upstream of the first and downstream of the third, just to save some time and resources. And we did use the YSI monitor and then also field kit, just the standard like Iowa Water or Snapshot chemical components. And it seemed that almost everything was didn't vary much from the field kit. Temperature and dissolved oxygen varied seasonally, but within normal ranges that we'd expect. Nitrate, and orthophosphate ammonia, all non-detect or really low. And then chloride varied, but it didn't seem to relate to anything else in the system. And this was early data just from 2000 through 2022. And it shows overall that there's close to a 30% reduction in E. coli numbers. I do have a graph later that has more recent data incorporated with it, but it's not in the same format. I apologize. I don't know, Steve, if you wanted to talk at all about the reduction. We do have a few spots where it did increase. And that could have been to any number of things or animals or other bacteria sources.
Yeah, there are some areas, there are definitely some times where you did see an increase for sure. And a lot of that depends on flow, right? Definitely some of those are low flow times. As you can see in some of those pictures, what you get behind the structure is a pool, right? A pool that backs up to the elevation of that filter. And the purpose is you're not going to get all the flow of that stream through the filter. Some of it's going to go over. You want some of that to go over. You don't want things to back up, and you also want transference. You want to be able to have transference of animals back and forth. But you're hoping that you have enough filters that you'll at least get a good chunk of that total water volume. It's hard to say. Treated, through that filter. So there's some portion of that water is flowing actually through that pancake of filters and getting treated in some way by some mechanism. right? And so when you have like in your late July, August, September, the low flow times, there's just not enough water to even get past the filters at times, or it's very low. We did notice that these E. coli concentrations were opposite, right? And so I think things backed up and just accumulated, and then the next time they balance back out. So, but we counted all the data, and just kind of things. That's why I kind of look at overall where we were, I think in some of the newer data, our decreases are not quite as sharp, and we're looking at, does this mean the filters may be getting clogged up a little bit? There is definitely sediment building up behind. So it's like I said, this is an experiment. What is there's no standard operating procedure. There's like, this is going to last 5 years or 10 years. We are learning as we go. So maybe it's a 7-year thing, a 5-year thing. We don't know at this point. We're just kind of trying to analyze data as much as we can. But we did notice over those first 3 or 4 years, yeah, this is showing a decrease in lab calculated E. coli concentrations. So I don't know what your next slide is,
Oh, yeah, we
Amy.
didn't do too much review.
Are you talking about goose?
There we go. So our second project is in the same area, but different watershed. And we just chose the number 6 this time to see if there was going to be a marked difference. And then they're more tightly spaced than Robin Creek. And this is it's close to 1,000 acres that are draining to this site, so quite a bit bigger than the first site. And there's a fair amount of ag, and then some light industrial. So it's a different type of different kinds of flow, and we did learn that there's different types of sediment. I've got a couple pictures to illustrate that. This was installed the same method. You can see on the right where we've got our notch in the middle. We left it maybe 14 inches or 18 inches, and then just anchored it down in the middle with those additional stakes. So you can see in these 2 pictures, there's a couple inches, maybe even 3 inches at the top of those stakes that are sticking out. And it's acting like a snag would traditionally, and it is kind of catching things up. So I wasn't out at this install too much, so I don't know if it was a completely different type of anchor or if they just made a judgment call on site. But I would, if any other practice went in, I would leave the least amount possible of that top of that stake sticking up. So here's a current condition from the install that we did in 2020. It's really hard to see, but it's kind of right in there. You can sort of see, lines that are perpendicular to the stream edge. So it's built up quite a bit. This was what they looked like originally, right? So this is, 5-6 years ago, and so they might not be functioning like they were initially because of all of the buildup. That's again, like Steve mentioned, one of the things that we're trying to find out. And then here's that's the second project site, current conditions. You can see on the left, there's just things getting caught up on the stakes, which isn't necessarily problematic. It depends on what it is. I think there was a shirt out there the other day, everything gets into the water somehow. And then this one, if you can sort of see that spot right there, is another one on our second project location. So that's been completely silted in. There are, I believe, 2 at the far end that have completely silted in. And it was a struggle to find them, but you can sort of see the end of that stake there. I mentioned that this site has a lot of ag draining to it as opposed to suburban developed area. And so there's a lot more sediment movement, and that wasn't really something that we took into consideration when picking the site, but we certainly will in the future. Just pay attention to the larger watershed area so we don't go through the efforts of install to have them silted in and like about a year and a half's time. And then here's the graph that's got all of the data from when we started up to the end. There's blank spots around, is it June 2025? And we had some of the lab data got botched up. I'm not exactly sure how, but just didn't come through. So we have a space there. So this is on the Robin Creek, the first install. So the blue is the site one, so upstream of everything, and the orange is site three, downstream of the three filters. I did take out two outliers here that were in the hundreds of thousands, just so we could see the bulk of the data more clearly. I don't know what, there must have been an animal or a new restroom spot found by the raccoons or something on those particular 2 days. So on those, I don't think we had much removals at all. But overall, we're still seeing trends downward. I think we're down to about, with all the data together, instead of that 27% reduction, we're down closer to a 12% reduction. So that might be that that function over time may be decreasing. They're fairly spread apart on the first project, so there could be even more factors that we're not incorporating. But Steve, I don't know if you had anything to add.
No, I think that's kind of where we are. We're looking at, and this is kind of a segue, I think, into talking about Augustana.
Yeah. Yep.
One of the things that when I was talking to Franklin is they were really hoping that we could help figure out what was going on. They knew it did something and they didn't know why, right? And we kind of had it in our ideas, but we're not microbiologists, and so that's kind of where like we see these reductions and or maybe sometimes like where we're reaching an end time, or how do we do this better? Well, for us to do this better, we have to really understand the mechanism of why it's working, and that's why we already work with Augustana a lot and have a great working relationship with everyone there. So we thought let's bring them in and some of their expertise to help maybe understand what's going on within these filters that's causing this reductions that we do see, right? What's the mechanism that behind this, so we can either enhance this, understand what we're doing, we could do better. So I'm not sure what the next slide is, Amy, but
Yeah.
Kimberly, this might be a great
I think Kimberly's
segue for you. Yeah, perfect
Yeah.
time for Kimberly to take over.
Yeah, if you want to give a brief introduction, Kimberly, and then kind of just talk about what the current project is, different components, that would be awesome.
Sure. I'm going to stand up and shut my door real quick. It's in between class, I realized.
Sure. So the city of Davenport has worked with the Upper Mississippi River Center through Augustana for many years on a lot of different types of projects, but it started and it has focused on watershed assessments primarily. They've also done some social surveys in regard to people's relationship with the stream near their house. They've also done some microplastic assessments and some other things, but this new study is just getting started, just getting underway, with Kimberly Murphy kind of leading the group there. If you want to talk about your work.
Yeah, hi folks. Thanks for allowing us to just talk briefly about what we've been up to so far and just some very, very, very preliminary findings. So I have a couple of research students this is their main project. Well, Caleb is the main student, but he really, when I was explaining this project to him, he really became interested in two things. One, the mechanism, like Steve mentioned, like if we're noticing a decrease in E. coli and total coliforms, like why, right? What's going on? And then he's also really interested in just like the interplay between like human health and the environment, and also just really stepping back and looking at the environment as a whole that's maybe around the filters. And so he has basically started the project about 3 weeks ago to do kind of a quantitative and a qualitative study around these filters, the biofiltration systems. And so he basically, for the month of April and May, he's going out once a week, and I go with him, and he's doing your basic water quality, water chemistry test. Amy mentioned the YSI, nitrates, phosphates, and then he's also running, tests for total E. coli and fecal coliform levels. He is basically starting most upstream from the first filter and then actually collecting samples in between every single filter, and then a final sample most downstream. He's also really kind of, again, stepping back, taking some qualitative notes. What's going on? Are we noticing extra buildup? Like at first we couldn't find the filters. Like Amy said, we found a shirt and of course a bunch of other trash. He's noticed things like in between some of the filters, there tends to be more raccoon trails. We had a bunch of ducks out the other day, which he was like, oh, that would play a role in the E. coli and coliform levels. And so he's starting to put all of this together. The other part of it is, again, coming back to this why, the mechanism. He had been reading in the literature about bacteriophages, which are basically viruses that, if you want to think about it this way, viruses that would remove the bacteria from the water. And so, I mean, there are tons of different bacteriophages out there. Each one basically has a different set of bacteria that it likes to kind of prey on, if you want to say it that way. And so, which ones might be there, we don't know, but are there any there, right, to begin with? And so he is basically running plaque assays, which allow for you to look at are bacteriophages present, and then start to quantify those. It doesn't allow for you to say which bacteriophage are present. That requires further DNA sequencing, for example, or RNA sequencing, but at least to get an idea, are they there, right? And so I touched base so we've been out there 3 weeks now, and bless his heart, he's been trying to get all the data entered. And so I said, I know it's the weekend, but could you just give me a quick, brief summary? And basically, as Amy mentioned earlier, for the YSI and the some of the other parameters, nothing really jumping out as like, whoa, that seems off. From what might be more, normal, within normal range. But he did note that he said, I am seeing decline. I'm just going to read what he said. I am seeing declines in overall E. coli and coliform numbers from most upstream to most downstream in both locations. However, Robin seems to be show more variability and noticeably, he calls it noticeably higher biological activity, which I take to mean more like the difference is more pronounced, I guess. And he also just said that, he is starting to realize, like, now as he's putting things together, he wants to definitely capture some more pictures and take some more qualitative notes. As Steve mentioned, you imagine the full span of the filter, and the water isn't going to flow over all of it or flow through all of it. You might have a section of it where it's very clear the water is flowing over it, and maybe the other section, there's just not enough water to flow over that section, or maybe it's more clogged up, if you want to think of it that way. The other thing that Caleb is looking into, or has started to, is what have others done to improve biofilter setups like this, right? And there's not a lot of these situations, these setups across the United States, to be able to, dig into the literature and have tons of articles to go by and previous research. But I know he mentioned to Amy and Steve that one group had used like carbon filters. So think of like the filters you use in a Brita filter, right? And it seemed to enhance the effectiveness of the biofilters by introducing some carbon filters. But, as Steve said, this is a scientific experiment. You go in with an idea, with a hypothesis, and you do some testing, you see where you're at. you make some notes and start to realize like, oh, maybe these do have a life, a shelf life to them. Um, so that's where we're at. Happy to answer any questions if you have them. Amy and Steve, is there anything else you would like for me to speak about?
I think that's it for now, since we just kind of got things started off.
Yep,
But
yep.
yeah, we're very excited to kind of add this type of information to our overall investigation. So
Yeah, I guess I will add too that we're also really excited to be able to take samples in between the filters too to see,
yeah,
maybe it's only one filter instead of three that you need, right? You notice that decrease after just one filter. So that's something else we're going to keep an eye on.
yeah. Great. Okay, I think I might just have a couple other slides. So these were our project partners to start off with. The second project, Davenport, we just did in-house without grant funds. And so we just, again, have that other role. So we're going to try to find another location to get some in either this year or next. Um, and I think that's all we have for you. There's a way to get to the Upper Mississippi River Center through Augustana if you want to scan that code, or the Partners of Scott County Watersheds website. I believe we have some information on the project on the website as well. But I'd have to go and see when the last time that was updated. I don't actually recall. But yeah, if you've got any questions for anybody today, we could entertain those.
Great. Yeah, thank you to everyone, Amy, Steve, Kimberly, for sharing the update on that project. I was really excited to hear about it because I used to work with these guys. So I was really excited that they were able to present to everyone here in Central Iowa today. So yeah, if you have any questions, it's a small group. So if you'd like to unmute yourself and ask, feel free. Just know that it's going to be on the recording. Otherwise, you can type your questions in the chat.
Yeah, we were discussing before we got started today that we think the project just the first one got started, and then Cassie got a different job because she was, yeah, the Partners Scott County Watersheds coordinator during first install. And so, it was crazy that it has been so long.
Yep.
And we're happy to share out results too when we get reporting from Kimberly and the students on their findings. We can get that sent over.
All right. Well, I don't see any questions. So we will go ahead and close out the lunch and learn. But once again, thank you so much for presenting to the group today. And I'm going to share this recording on the Rain Campaign website. If you can't find it, just email me and I'm happy to email you a direct link and so you can share it. All right. Thanks, everyone. Have a great day.
Yep. Thanks for having us,
Thank you
Cassie.
all.
Thank you.